Good morning/afternoon, everyone. Today we are going to walk through one of the most critical frameworks in pharmaceutical quality assurance, the ICH Guidelines on Pharmaceutical Impurities, specifically Q3A, Q3B, and Q3C. Now, why does this matter? Every drug product contains more than just the active ingredient. During synthesis, manufacturing, and storage, small amounts of unwanted substances, what we call impurities, inevitably find their way into the final product. The question is not whether they are there. The question is, are they safe? That is exactly what the ICH Harmonized Framework answers. It establishes rigorous, internationally agreed standards for identifying, reporting, and limiting these impurities, based on their potential risk to human health. So let us start from the beginning. Classification and sources of impurities. First, let us understand what kinds of impurities we are dealing with and where they come from. The framework recognizes two broad categories. 2A, organic and inorganic impurities. Organic impurities are the most common. They arise from two main sources. Synthesis side reactions during the chemical production of the drug substance can generate byproducts. Degradation. The drug substance or product can break down over time, especially under heat, light, or humidity. Inorganic impurities are different in nature. These include catalysts used in chemical reactions, heavy metals from equipment or raw materials, salts and other inorganic residues. These can be process-related or come from the manufacturing environment itself. 2B, degradation products in drug products. A separate and important category is degradation products, and here we are specifically talking about the finished drug product, not just the active substance. These arise from interactions between the drug and its excipients, or from the container and closure system during manufacturing and long-term storage. For example, a preservative in a liquid formulation might react with the active ingredient under certain temperature or pH conditions, generating a new chemical entity that was never intended to be there. This is why stability testing is so closely linked to impurity profiling. 2C, residual solvents, 3 classes. The third category is residual solvents, organic solvents used during manufacturing that remain in the final product at trace levels. ICH Q3C classifies these into 3 classes based on their toxicological risk. Class 1, avoid. These are solvents with known or suspected human carcinogenicity or severe toxicity. Examples include benzene and carbon tetrachloride. Their use should be avoided unless absolutely necessary, and even then, only at extremely low levels. Class 2, limit. Solvents with non-genotoxic animal carcinogenicity or possible irreversible toxicity. These must be used below established limits. Examples include acetonitrile and methanol. Class 3, low toxicity. Solvents with low toxic potential to humans. They are acceptable at higher levels but should still be reported if present above 0. 5% of the drug substance. The visual of the 3 beakers, red, yellow, and green, is a helpful way to remember. Red means avoid, yellow means limit, green means lower risk but still control. Section 3, Thresholds and Safety Qualification. Now we move to the practical heart of the framework. What do you actually do when you detect an impurity? The answer depends on how much is present, and this is where the threshold system comes in. 3A, the 3-tier threshold system, ICH-Q3A. ICH-Q3A establishes thresholds for new drug substances, and it differentiates based on the maximum daily dose the patient receives. The framework splits products into 2 groups. Those with a daily dose of less than or equal to 2 grams per day. Those with a daily dose of 2 grams per day. Why does the daily dose matter? Because the absolute amount of impurity a patient is exposed to depends directly on how much drug they take. Higher daily doses mean even a small percentage of impurity translates into a larger absolute exposure. Let us go through the 3 threshold levels. Reporting threshold, this is the lowest level. If an impurity is detected above this concentration, the manufacturer must simply report it in their regulatory documentation. For products less than or equal to 2g/day, 0.1% or 1 mg per day, whichever is lower. For products 2g/day, 0.05%. Identification threshold, if the impurity exceeds this level, the manufacturer must structurally identify the impurity, that is, determine what the chemical entity actually is. For less than 2g per day, 0.15% or 1 mg per day. For greater than 2g per day, 0.05%. Qualification threshold, this is the highest and most demanding level. If an impurity exceeds this threshold, the manufacturer must qualify it. Meaning they must conduct biological safety studies or provide scientific literature to demonstrate that this level of exposure is safe for patients. For less than 2g per day, 0.15% or 1 mg per day. For greater than 2g per day, 0.05%. So to summarize the cascade, detect, report, identify, qualify. Each step demands more scientific rigor. 3b, permitted daily exposure, PDE. A key concept in this framework is the permitted daily exposure, or PDE. The PDE is a pharmaceutically acceptable intake level, a calculated value that represents the maximum amount of a residual solvent or impurity that a person can be exposed to every day over a lifetime, without any appreciable risk to health. It is derived from toxicological data, animal studies, human data where available, and incorporates safety factors. The PDE is essentially the scientific backbone that gives meaning to all those threshold numbers. Think of it as the bridge between raw chemistry and patient safety. 3c, the safety qualification process. Finally, let us look at the safety qualification process. What happens when an impurity exceeds the qualification threshold and we need to formally demonstrate it is safe? The process follows a clear sequence. 1. Data collection and evaluation. All available data about the impurity is gathered. Analytical data, literature, toxicological studies, or new studies commissioned specifically for this impurity. 2. Biological safety established. Based on the data, a conclusion is reached. Is the biological safety of this impurity established at the observed level? If yes, the impurity profile is accepted. If no, the manufacturing process must be modified to reduce the impurity below the threshold. This is not a one-time exercise. It is part of an ongoing lifecycle approach to pharmaceutical quality, the same spirit we see in process validation under ICH Q8, Q9, and Q10. Thanks for watching. If you found this video helpful and want to keep growing your knowledge of GMP, pharmaceutical quality, and compliance, then make sure you hit subscribe and turn on the notification bell. I upload clear, practical, and easy-to-understand videos on GMP topics that help you stay confident and compliant in your daily work. Your support helps this channel reach more professionals who want to learn and improve. Stay curious, stay compliant. And I'll see you in the next video.